Well, it's a real honor and a pleasure to introduce Susan Lindquist to this audience. She obtained her doctoral degree from Harvard University, although we don't hold that against her. And she was the Lasker Professor of the Medical Science at the University of Chicago before she moved to the Massachusetts Institute of Technology in 2001. She's been a member of the Whitehead Institute there, and she served as director from 2001 to 2004. Her research focus has been on protein folding, and she's really led this field into multiple new directions. She's shown that protein conformational changes are critical in a range of effects: cellular stress, evolution, transmission of traits, and most recently neurodegeneration, which I think is going to be the focus today. In one of these lines of work, in terms of the role in phenotypic variation and evolution, she showed specifically that HSP90 can buffer silent polymorphisms, and when that function is lost, these can be manifest and be selected, and genetic diversity can be evolutionarily advanced. She's also shown that HSP40 can be involved in clearing protein aggregates. In terms of transmission of traits, she's also made groundbreaking discoveries, the second whole line of work. There had been the idea of protein conformation, transmission of traits, but it was Dr. Lindquist's studies that really, using yeast genetics, that brought this home and provided the most convincing evidence that protein conformations can transform without nucleic acids. Finally, in the last decade, she's focused on neurodegenerative disease, which I think she's going to talk about primarily today. There'd been a lot of evidence that protein folding matters, but how this affects the cell and how it makes cells sick hadn't been clear. It's her ability to harness yeast genetics to reduce this problem to single cells that has really made tractable advances, and I think has the promise for the next century of research making real inroads in Alzheimer's, Parkinson's, beyond. She's won a number of honors, Dixon Prize, the Otto Warburg Prize, Max Delbruck Medal, the U.S. National Medal of Science, and she's a member of the National Academy. So let's welcome Susan Lindquist.
Thank you so much, and I want to thank everyone involved with this symposium for inviting me, because it's just been absolutely fantastic. I want to thank the previous speakers for a wonderful set of talks. So I work on a variety of different problems involving protein folding, and we haven't given up the HSP90 or prion stuff yet, even though we've started this. And basically the problem of protein folding is that every protein has to fold into these incredibly complicated, very, very specific structures, and each protein folds into a different structure. And they have to do that in an environment that's just absolutely absurd. Yeah, they have to do that inside of a living cell. And the reason why it's absurd is that the concentration of other proteins in that cell is just staggering. It's about 300 milligrams per mil. It's about the same concentration as the concentrations of proteins in packed crystals that crystallographers use to study structures. But of course, the proteins inside of a living cell are not static. They're moving around at actually incredible speeds. They're bumping into each other all the time. And so getting a protein, an extended polypeptide chain, folded properly, and maintaining those folds when they're colliding with each other with incredible kinetic energy is really a very, very difficult problem. And so when you think about proteins going about their business inside of a living cell, it's very much not like Esther Williams and her mermaids. It's much more like the wave pool at the Summerland Amusement Park on a really hot summer day. So just imagine if a little bit more kinetic energy were put into that system, for example, with the heat shock, or if a little bit of the water was taken out of that system, for example, with an osmotic stress, or if some of the individuals in that see, we're not really as strongly structured as other individuals. You can imagine it would lead to a crisis in a hurry. And in fact, that's happening in cells all the time. Living systems are poised on a protein folding crisis. And the reason why they do that is that there's tremendous efficiencies to be gained from having proteins so closely packed together. You can pass electrons from one protein to another. But they pay a very high price for that because there's constantly this danger of proteins going off pathway and proteins misfolding. And in fact, we now understand that many, many human diseases are caused by problems in protein misfolding. And thinking about them that way might provide a line of attack for us. And so we've been studying this protein folding problem for quite a long time. And finally decided that, well, maybe we ought to tackle some of the human protein folding diseases. And these are just some of the diseases that involve protein misfolding. These happen to all be neurodegenerative diseases. Things like frontal temporal dementia, ALS, Parkinson's disease, Huntington's, Alzheimer's, prion diseases. And they're all caused by the misfolding, or appear to be caused by the misfolding, of different individual proteins in these different diseases. And so because we started to learn quite a bit about protein folding, we thought, well, maybe we should try to tackle these because the devastation that these misfolding processes wrought in a living human brain is just staggering. Any of you, this picture speaks for itself, but I'm sure most of you who have met or encountered people with this disease or had the misfortune of having your loved ones suffer from it will understand why we really need to tackle these diseases, and especially as our population is getting older and older. Perhaps I especially wanted to look at it as I started contemplating myself getting older and older. I really wanted to think that maybe we could start working on this. And the idea was to try using yeast cells. These are typical yeast cells. They're budding yeast, Saccharomyces cerevisiae, as a sort of a living test tube to study these complicated protein folding problems. And if your attitude is like these folks here, you're not alone, because this is very much the attitude we faced when we first proposed this. I had a very, very, very hard time getting any funding for this. I had a hard time getting anyone to work on it in my laboratory. But as Mike today made a statement to encourage people to continue working in the face of otherwise discouraging events, the student who tackled this has really first started to tackle this in my lab and has now led to many other people who've been willing to take it on, has actually found that, in fact, when we take these proteins into a yeast cell, they misfold and they're toxic, but they don't just cause a non-specific generalized protein aggregation toxicity. In fact, the kinds of toxicities we get for each of these proteins is very specific for each of those proteins, and it depends upon where that protein is misfolding and what other types of proteins it's interacting with when it misfolds. And the things we find in these yeast models of neural pathological processes, actually both genetics and compounds that we find matter to neurons that are suffering from the misfolding of these same proteins in very specific ways, and in fact connect up with all sorts of aspects of human diseases, both in terms of the toxins, the nature of the toxins that can be associated with these neurodegenerative diseases, and the human disease alleles that are associated with these diseases. So the reason, of course, for doing anything in a yeast cell is there's two. And one of them is that yeast cells are eukaryotic cells. Neurons are too. Neurons are very, very fancy eukaryotic cells, but they're eukaryotic cells. And all of these things are very, very highly conserved in yeast and in neurons. Lipid biology, vesicle trafficking and fusion, and the ways in which that's regulated, lysosomes and peroxisomes, autophagy, apoptosis, complicated cell cycle, mitochondria and oxidative stress, and of course, a whole realm of proteins that are involved in taking care of that problem I told you about, the protein folding problem. And the cell actually devotes about 5% of total protein in the cell to taking care of this problem. And it involves chaperones, it involves proteasomes, it involves ubiquitin ligases, it involves autophagy and osmolytes and all sorts of mechanisms. Anyway, these are very, very highly conserved from yeast cells to neurons. And the other reason for wanting to work with yeast is that it has an absolutely unrivaled toolkit. I mean, there's a lot of great things we can do with mice now and nematodes and fruit flies, but believe me, there is no organism on the planet we can manipulate as well or as facilely as we can yeast cells. And in fact, the amount of information we have on protein-protein interactions and genetic interactions is unrivaled. And furthermore, they're single cells. Now that may seem like a really crazy thing to think of as an advantage when studying a neurological pathology. But I'll tell you why I think it's a very big advantage. Neurons are talking to each other and are talking to glia cells and their support systems all the time. It's talk, 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 talk. And their interactions with each other are profoundly influencing them. And so if you want to try to actually study the fundamental underlying process that's responsible for these diseases, and we have no therapies, not one, currently aimed at the fundamental underlying problems that are going wrong with these proteins. If you want to try to study that, it would be nice maybe to isolate all of the complexities of these interactions with other proteins and other neurons and other cell types, and just really look at the problem of that particular protein in an individual isolated cell. So I'm going to tell you about two of these pathologies that we've been looking at. One is involved with synuclein, and the other is A-beta. The synuclein work we've published most of, although we have a lot more stuff that I'm not going to tell you about that hasn't published. But the A-beta story has been submitted but not yet published. So the synucleinopathies are varied: Parkinson's disease, dementia with Lewy bodies, and multiple systems atrophy. And they're all characterized when you slice through a brain, as I showed you those sections of pathologist slides earlier, those brown blobs that were protein aggregates. Well, these synuclein aggregates can occur in different cell types, and when they occur in different cell types, they're associated with different diseases. But certainly the one that we know the most about, and the one that is the most common, is Parkinson's disease. And Parkinson's is really a spectrum of disorders. It's the second most common neurodegenerative disorder. And it's particularly characterized by the loss of particular types of dopaminergic neurons, which lead to severe motor defects and more. But it's now appreciated that the pathology is broader than just the dopaminergic neurons. It's been it's a very complex and bewildering disease because there are a lot of associations with environmental toxins, such as, for example, manganese. Miners of the metal manganese get PD twice as often as miners of other metals. And recreational drug users, where the manganese has contaminated the drugs, have gotten the disease. And there's also a variety of mitochondrial poisons that are associated with PD, or at least Parkinsonism. And then there are many different genetic factors that contribute, and these are called Park loci, and they're being elaborated more and more all the time. And many of these diseases, even though they're associated with different genetic lesions, when people go to autopsy, they see the Lewy bodies, or this alpha-synuclein aggregation, not by any means in all of them, however. And finally, Park1, the first Parkinsonism allele, is a protein called alpha-synuclein, which is in those aggregates. And it was also found that mutation of alpha-synuclein, or simple amplification of the wild-type protein, is sufficient to cause early onset Parkinsonism. Now, this one is a relatively rare cause of PD. This one is not so rare, but still, the very fact that this particular protein is aggregated in this disease, with all these different environmental and genetic causes, and the simple overexpression of that wild-type protein is sufficient to cause the disease in humans, made us think that it might be worth trying to overexpress that protein in a yeast cell and see what happened. And we were encouraged by another thing with regard to trying this, and that is that alpha-synuclein is a very simple little protein. It's not that like the LERK kinase, which some of you may know is also involved with PD, which is a very complicated multi-domain protein. Alpha-synuclein is a 14 kilodalton little tiny protein that folds into 4 alpha helixes. It associates with phospholipids and membranes. And when it's not associated with membranes, it's natively unfolded, and it's prone to forming oligomers and larger polymers. And these are believed to be the toxic species, and especially the oligomers, although don't take this structure as being serious. It's just an imaginary structure. But the oligomeric forms of the protein that accumulate when the natively folded protein is not associated with membrane are believed to be part of the toxicity. So we put alpha-synuclein stitched up to a green fluorescent protein on a very easily regulated promoter in yeast, so we can normally keep it off and turn it on whenever we want to. And when we put in 2 copies of that gene in a yeast cell, it goes to the plasma membrane, and that's great. That was just what we expected it to do, because in human neurons, it's localized to vesicles at the ends of synapses. And one difference between yeast and neurons, and there are, I will admit, quite a few, is that in yeast cells, secretion process is constitutive. So if alpha-synuclein was getting onto secretory vesicles in a yeast cell, what we would expect it to do is just wind up on the plasma membrane. So if we express a little bit more alpha-synuclein, or a little bit more alpha-synuclein, the localization of the protein changes profoundly, and the viability of the cells changes profoundly. So these cells grow perfectly normally, these cells grow slowly, and these cells die. And so that provides us very immediately with a platform for doing genetic screens. And in collaboration with Josh Lebert, who did almost all of the work, we just helped him finish making a library of the yeast genome that contains every open reading frame of the yeast genome under the control of the same promoter that we use to induce alpha-synuclein. And we like this library a little bit better. Many of you may be familiar with the yeast deletion library, and that's been a great system too. But the yeast deletion library is a bunch of strains that have been coping with deletions of various proteins for long times, and all kinds of changes have taken place in those cells. With this overexpression library, what happens is we transform a cell that has a moderately toxic level of alpha-synuclein with each one of these plasmids, assemble a library of strains that contain those different plasmids, and then we husband them on glucose so the cells aren't experiencing anything, any change. And only when we transfer them to galactose does the alpha-synuclein gene and the other gene turn on at the same time. And so we can test how very cleanly how that gene modifies the toxicity of alpha-synuclein, looking for either enhancers or suppressors. And so more recently, we've gotten robots that allow us to do this much more efficiently, and this is how our current screens are looking. These are robots that have printed our various yeast strains onto media, and these cells are not induced and they're growing just fine. These cells are induced and they're growing more slowly. And what you'll see is there are groups of 4 here, and that's because we test every gene 4 times, and it gives us much more reliable results. So we print all 4 of these strains contain the same test plasmid. All 4 of these strains contain the same test plasmids. And this is clearly an enhancer of toxicity. It makes things worse, and this is a suppressor of toxicity. So then, of course, the things we get out, we have to test in a neuronal system. And we have tested them in 3 different systems. We've tested them in C. elegans in collaboration with Guy Caldwell, in Drosophila in collaboration with Nancy Bonini, and in primary rat embryonic midbrain cultures in collaboration with Chris Roche, although we now have this up and running in my own lab. And we are beginning what I think is going to be a very exciting collaboration with Rudolf Janisch to translate our things into human iPS cells derived from patients, because these diseases are very complicated. There are a lot of different underlying genetic factors, and I think for any gene disease that's going to need personalized medicine, I think this is a real poster child. Anyway, do our genes that we got out of a yeast cell that have conserved human homologs actually matter to a neuron expressing alpha-synuclein? And so I'm just going to tell you that the very first gene we got out that had a very strong effect was a Rab GTPase that's involved in controlling vesicle trafficking. And it controls vesicle trafficking from the Golgi to the ER, and is a very closely related Rab GTPase that in neurons have more of these things than yeast cells do, that in neurons controls vesicle trafficking at the end of the synapse. And so I'm going to just tell you that this Rab1 worked in nematodes and it worked in fruit flies. I'll just show you the results with primary dopaminergic neurons from the midbrain region of the embryonic rat that were put into culture. And we get a rough sense of how many dopaminergic neurons there are in these cultures by staining with TH. And in these cultures, we typically get about 7% of the neurons are dopaminergic neurons. And when we express alpha-synuclein, and here we're expressing a mutant that's associated with early onset PD, it kills actually lots of the neurons in the culture, but it kills the dopaminergic neurons even more frequently than it kills other neurons. And so the percentage of the dopaminergic neurons goes down. And if we co-express LAC-Z with that, it has no effect. But if we co-express that Rab1 GTPase, it has a pretty strong rescuing effect in these neurons as well as it did in yeast. We also see changes in vesicle trafficking, and in fact, subsequent experiments, I'll show you, we find that we can directly assess the effects of alpha-synuclein on vesicle trafficking. So we've done this now with 9 different genes, which have very diverse functions, so functions that you wouldn't necessarily think would be connected, but they're clearly connected because they modulate the toxicity that's caused by overexpressing alpha-synuclein. And 8 out of those 9 genes that we tested from our yeast screen and tested in the neuronal system have worked. So I mentioned that vesicle trafficking seemed to be particularly sensitive, and in fact, when we looked at our yeast cells that were accumulating alpha-synuclein and where it was accumulating in a toxic state, we started to see vesicles being blocked, but they were not fusing with the target membranes. And Virginia Lee actually has done autopsies on human patients with PD now, and she's seen that, in fact, the Lewy bodies are associated with a lot of trapped vesicles. So what does alpha-synuclein do in vesicle trafficking? Well, Charlie Barlow has this wonderful assay in which he isolates ER, and he can examine vesicle budding, and in the addition of cellular factors, he can examine docking and fusion by the fact that when the vesicles dock, the proteins get modified. And so we collaborated with Charlie and found that, in fact, what alpha-synuclein does is it doesn't interfere with vesicle budding at all. In fact, if anything, it stimulates it. But when it comes to vesicle fusion with the target membrane, it completely blocks it. And in fact, alpha-synuclein is associated with that focus of blocked vesicles. When I showed you in those earlier cells, those big kind of green bowling balls of GFP-labeled alpha-synuclein, when you look at those by electron microscopy, they're blocked vesicles with high concentrations of alpha-synuclein surrounding them. So alpha-synuclein causes problems specifically in vesicle trafficking and at specific stages of vesicle trafficking. We've also found one of the genes that we found that suppresses the alpha-synuclein toxicity was annotated in the yeast genome as being yeast open reading frame 291W. Not very informative because nobody even in yeast knew what this was doing. But it was of interest to us because we found that it is very, very highly homologous to a human protein. And moreover, about 6 months after we found it in yeast, this paper was published that hereditary Parkinsonism with dementia is caused by mutations in that protein. And it encodes a lysosomal protein that's a P-type ATPase. So it's at the lysosome. It's in a different place from the alpha-synuclein in the cell, and there would have been no reason from the human data to connect it up with alpha-synuclein toxicity. But we find it as a suppressor. When we overexpress this function, we find it as a suppressor of alpha-synuclein toxicity. We were able then, and I'll spare you the details, we're able then to find that this protein, what it's doing is it's a P-type ATPase, and what it's doing is it's pumping manganese. And it pumps manganese into the vacuole to reduce the cellular concentrations of manganese. This is very interesting because remember I told you earlier that there's been associations between manganese toxicity and Parkinsonism. And interestingly, we had 60 metal ion transporters in our screen We got out 3 metal ion transporters that suppressed the alpha, or one was an enhancer, 2 were suppressors of alpha-synuclein toxicity. All 3 of them are manganese transporters. One of them transports manganese and calcium into the ER and affects Golgi trafficking. And another one of them transports manganese and iron. And all 3 of those metals have had very interesting history of associations with Parkinsonism. Our microarrays also told us that there's a lot of mitochondrial damage and that alpha-synuclein accumulation in these aggregates is causing bursts of reactive oxygen species. So we then did a chemical library, the screen, for things that would suppress. And what we did was we screened 150,000 compounds over at the Harvard ICCB. And we asked, in this case, for restoration of toxicity in that really high toxicities, amelioration of toxicity and that really high toxicity strain, not the intermediate toxicity strain, which we screened genetically, because genetically we obviously found it was very complex. We're finding all sorts of different things going wrong. And so we wondered if in the higher toxicity strain, when lots and lots of things are going wrong, mitochondrial toxicity, all sorts of things are happening, would it be possible to find a compound that would rescue against that? It was a very rigorous screen, a high bar to pass. We found some compounds that did. We went back and looked at vesicle trafficking, and it turned out that they fixed vesicle trafficking. They also fixed the mitochondrial defects. And they work in the nematode, and they work in rat neurons in culture too. So this is an example of a test done in nematodes, and it's actually not easy to get drugs into these little beasts. They live out in the soil, and they have lots of drug pumps, and so you have to do things like dissolve the drugs in DMSO and all kinds of yucky things, but you can do it. And basically what we have here is we've got alpha-synuclein being expressed in the dopaminergic neurons of nematodes. Nematodes don't have a very sophisticated nervous system, but what they have is dopaminergic neurons, and they always invariably have exactly the same number of dopaminergic neurons in exactly the same position. And so in this particular view, we always see 4 dopaminergic neurons in wild-type animals. When they're expressing alpha-synuclein, some of the neurons die, some of them just don't extend their processes. Actually, this is an age-related phenomenon, so they actually start out looking good, but then they regress. And if we treat the worms with one of the compounds we got out of our yeast screen, the neurons get better. And finally, we were able to take, as I mentioned, there are mitochondrial poisons that kill neurons in ways that are believed to be very closely related to Parkinsonism. And in fact, you can create diseases in animal models with these kinds of compounds. And in fact, humans who have accidentally encountered these compounds can get Parkinson's disease. And these are mitochondrial poisons. And this just simply shows you that a dish of neurons that looks like hell because it's been exposed to one of these mitochondrial poisons. And this is what these neurons look like when they've been exposed to the mitochondrial poison in the presence of our compound, which we got out solely by its ability to rescue yeast cells from alpha-synuclein. So I think the fact that we're able to get out both genes and compounds that matter to neurons and matter to the toxicity of alpha-synuclein in neurons indicates that it's not that alpha-synuclein is killing dopaminergic neurons because there's something completely unique to those dopaminergic neurons. You don't have to only study the pathology process in a neuron, that it's actually alpha-synuclein is causing some fundamental process that's interfacing with very, very conserved basic aspects of eukaryotic cell biology. And the question then is really, why does it hit dopaminergic neurons? What are the special vulnerabilities that make dopaminergic neurons particularly sensitive to this general pathology? And I think that there are many of them, and we're only beginning to understand it. But certainly, of course, neurons are very, very highly dependent upon vesicle trafficking, especially of neurotransmitters. The foci of alpha-synuclein, at least we find in a yeast cell, seems to lead to a very high local concentration when those vesicles get blocked. The protein comes on and off the membranes, and then you start to form toxic oligomeric species, which do other types of damage to the cells. There's some special vulnerability to metal ions, which we don't yet understand, but what we can say is it's related to the misfunction of alpha-synuclein. Neurons have very, very high rates of respiration, very, very dependent upon mitochondria, especially dopaminergic neurons. And finally, dopamine is in particular a particularly dangerous neurotransmitter because it's made out in the cytoplasm, and it needs to be pumped into vesicles to prevent it from becoming toxic and decaying and causing all kinds of reactive oxygen species. And in fact, so if you have a defect in the production of vesicles, in the forward trafficking of secretory vesicles, a dopaminergic neuron would be more sensitive than many neurons to that process. So I'm now going to turn to something different. I'd like to tell you about what we've been doing with A-beta toxicity. But before I do, I'm going to first have a very brief foray into prions. And you'll see why, because there's a relationship between prions and A-beta that I think is important. So Eric talked about prions yesterday, I understand. And this is an animal suffering from the prion disease known as scrapie. It's a disease that's caused by the misfolding of a protein. The protein is actually the infectious agent in the disease. It misfolds and causes other proteins of the same type to misfold too, and that can be spread as an infectious agent. Turns out that yeast cells have prions as well, and my lab has worked quite a bit on these. These red cells and these white cells are genetically absolutely identical. The difference between them is that these white cells have one particular protein in an altered prion conformation. And that alteration in protein folding is self-perpetuating. It's self-templating and self-perpetuating. And so it actually serves as an element of genetic inheritance. These cells inherit this in a very, very orderly way. And there are actually systems. The reason I got into this is because the protein I've been working on called HSP104 is actually a device that's employed to partition these aggregates into daughter cells and make sure that the cells inherit it in a very orderly way. Now, this doesn't just change cells from red to white. It creates all kinds of wonderful phenotypes in these cells and does all kinds of interesting things. We actually have found 25 prions in yeast, and I know Eric conveyed to you the message that prions can be great. They're not just terrible. And we think prions are great in yeast, and I'm not going to go into any of that. I'm just mentioning prions for one reason. These are little foci of prion aggregates that you can see being inherited by the mother cells to the daughters. And what those foci really are is amyloid fibers. And of course, amyloids figure heavily in all of these different neurodegenerative diseases. And we found that if we could purify this protein in the soluble form, it would assemble in a test tube, slowly, very inefficiently. But if we took a very small bit of the pre-assembled protein and added it to the start of a new reaction, it would do that. So the ability of this protein, amyloid, to self-template itself is extremely, extremely efficient. And that's what allows you to have these two different states. It's hard for the protein to get into the amyloid state, so mostly it's soluble. But when it gets into the amyloid state, it stays in that state and gets passed on and inherited. Well, very early on, years ago, we found that there was a very odd mechanism of polymerization for this protein. It assembles into an oligomeric species, a molten oligomeric species. It's a natively unfolded protein, rather like alpha-synuclein. If you purify the protein and put it into physiological buffer, it's natively unfolded. Forms these molten oligomeric species, and they then assemble into amyloids. And shortly after we published that paper, it was the fact that A-beta assembles in a very similar way was described. And then Charlie Glabe did something amazing. He made an antibody against the oligomeric form of A-beta that does not recognize the monomer or the amyloid. It recognizes something about the conformation of the oligomer. And oddly enough, it recognizes something about the conformation of oligomeric species seen with alpha-synuclein and several other proteins that are associated with neurodegenerative disease. So there's some common conformation in this oligomeric species, which many people now are beginning to believe is really the most toxic form in all of these diseases. And I call it Charlie's magic antibody because if you take reactions out of our yeast assembly at various times, this yeast prion, which has not anything even remotely related in sequence to the A-beta protein, that antibody does not recognize the monomers. It recognizes an oligomeric species that begins to assemble and very quickly converts to amyloid, and the antibody no longer recognizes the amyloid. So there is something about the conformations of these proteins that are natively unfolded and can get into amyloids where they dwell in an oligomeric species that has some mysterious property that's common amongst them. So why aren't the yeast amyloids that I'm telling you about, I'm telling you prions are good in yeast, why aren't they toxic? The reason is because they very efficiently assemble into amyloids. And in fact, if we, whoops, I'm supposed to be talking now, it looks like. They very efficiently assemble into amyloids, and if we do things that cause them not to assemble very well, they become very toxic. So the basic message then is that this problem in protein folding is as ancient as the hills. These same types of misfolding forms accumulate in yeast cells as well as in higher neurons. And so it might seem reasonable, not so unreasonable, that we could maybe take a look at the toxicity and the things that affect the toxicity of A-beta in a yeast cell. So how do we do that? Well, whoops. Yeah. Many of you, I'm sure, know that A-beta is actually made from a much larger precursor protein called APP that appears first, of course, in the endoplasmic reticulum and then traffics through the Golgi, goes into various secretory vesicles, winds up being secreted from the cell. A-beta can be secreted. The APP in its unprocessed form can come back in and be endocytosed. A-beta can be generated in endocytic compartments and in the lysosome. It's a very complicated trafficking, and no one really knows quite where all of the toxic stuff is, although certainly stuff out in the plasma membrane matters a lot. So how do we replicate this in a yeast cell? We don't want to deal with the processing of APP, so what we do is we take A-beta and put a secretory signal on it. So that gets A-beta expressed in the ER, and then it gets cleaved, and it has no retention signal on it. So it will start to migrate to the Golgi. It will go to secretory vesicles. It will get out to the plasma membrane. And here's an extra advantage of the yeast cells. They've got a cell wall. So that keeps the A-beta local. It keeps it at a high local concentration to the plasma membrane, and it doesn't just float away. And that allows it to come back in by endocytosis and appear in various compartments of this very vast secretory compartments of the cell. So we do an unbiased screen of 6,000 yeast genes for things that modify toxicity. And these are the genes that we found that have high, very close human homologs. We find 3 genes involved in Clathrin-mediated endocytosis. Very interestingly, this gene here is the 3rd highest hit on the human genome-wide arrays for alleles associated with risk of Alzheimer's disease. It's been replicated in 11 different genome-wide array studies in both sporadic and familial AD. This gene right here, there was a new study of Alzheimer's disease alleles in which popped out 2 additional genes involved in endocytosis. And this gene right here is the direct partner protein of one of those other genes which polymorphisms are associated with early onset AD in people. And we have also unpublished 2, actually there's a total now of 5 different genes in this gene list that look like they might be associated, polymorphisms are associated with early onset Alzheimer's disease. Some of them are still a little bit iffy, but 3 of them are pretty damn good. So we created to see whether or not we could duplicate the effects of these genes in a glutamatergic neuron, a neuronal cell type that's related to AD. We expressed A-beta in the glutamatergic neurons of the nematode. And what you see here, we've marked them with GFP, and you see a certain number of neurons all the time in those worms. But when they're expressing A-beta, you see fewer of those neurons. And it's an age-related thing. So at 3 days, only about 50% of the worms have the normal number of neurons. At 7 days, only about 30% have the normal number of neurons. So this is an age-related decline. And if we co-express M-cherry or LAC-Z, nothing happens. But if we co-express our endocytosis suppressors, they suppress. We co-express 3 other unrelated suppressors in different pathways, 2 of which have connections with human genome-wide arrays. They suppress. And if we express an enhancer, it enhances. So every gene we've tested in the nematode affects the toxicity of A-beta in these glutamatergic neurons in the same way as it does in yeast. Finally, we went to rat cortical neurons. There aren't very good models for studying A-beta toxicity, but one of the things that people do is they assemble the A-beta into oligomers and stick them onto neurons, and neurons seem to be dying in ways that might be related to AD. There hasn't actually been many very good demonstrations of that, but at least one gene, ApoE, has been demonstrated to affect the toxicity of A-beta applied in this way. And now we find that if cells are exposed to a vehicle alone with GFP or P-column, there's no difference. But if we expose them to A-beta oligomers, the cortical neurons are drastically affected, and GFP has no effect. But P-column rescues in a dosage-dependent manner. So P-column has been, although it's been very, very highly validated in these human GWA arrays for Alzheimer's disease, it's been thought to actually work by since it's a gene involved in endocytosis, it's been thought to work by affecting the trafficking of APP. But we're not expressing APP, we're expressing A-beta. So as we show here, it's directly involved in modifying the toxicity of A-beta in a yeast cell and in a neuron. So now we can return to yeast cells and actually ask what's the mechanism. There's two obvious things you can think about. One is, it could just be that the stuff is on the plasma membrane, that's bad. You want endocytosis and dump it into the lysosome or the vacuole to get rid of it. So you want to upregulate endocytosis. That's interesting, but it's not terribly sophisticated. But what if instead it might be that A-beta actually is specifically toxic to the endocytic machinery? And the reason why you need to upregulate endocytosis is to cope with a primary toxicity induced by A-beta. So to look at that, we looked at a membrane receptor that's normally involved in binding to mating pheromone on the surface of yeast cells, and in the absence of mating pheromone, endocytosis takes it and dumps it into the vacuole, and you can see that when you tag it with GFP. What happens when you express A-beta? The vacuolar trafficking of this receptor, plasma membrane receptor, is profoundly disturbed. Our modifiers restore, not completely, but restore significantly the trafficking of that receptor. And then, interestingly, these two genes should be working in rather different ways. They both work on Clathrin-mediated endocytosis, but one of them is involved in increasing the production of Clathrin-mediated vesicles, and the other is involved in getting those Clathrin-mediated vesicles to the next target, the target membrane, and uncoated. And in fact, when we look at cells in which we've monitoring the Clathrin itself, we see that A-beta screws up Clathrin trafficking, and one of the modifiers fixes that trafficking defect by increasing the amount of Clathrin-mediated coated vesicles, and the other one increases the flow to that pathway by increasing the uncoating of the vesicles. Anyway, this is just to finish up very, very quickly now. We've screened 120,000 compounds for things that will restore viability to yeast cells expressing A-beta, and we have gotten quite a few moderate strength suppressors. A few go as high as rescuing 50% of cell growth. Several of them also work in the nematode to rescue from the glutamatergic neuron-induced cell death of A-beta. One works in a mouse AD model and restores both, reduces pathology and restores cognition. And we haven't done that work. Actually, it turns out that our yeast screen has hit a compound that the laboratory of Ashley Bush and Rudy Tanzi have been working on. It's actually currently in clinical trials. It's actually the only thing currently in clinical trials that is believed to modify the toxicity of A-beta itself. And it's a compound that I don't think, it's got a long history of toxic effects and all sorts of other things. I don't think it has a lot of legs on it in the long term, because in fact also in our yeast cells, it rescues the yeast cells quite nicely, but it's a very delicate balance in how much of it you add, because it will then cause cell death. But we have a few compounds that actually have a fairly broad range where we can suppress toxicity and add them at tenfold higher concentrations, and they don't cause toxicity. So I'm hoping that this is just the tip of the iceberg. I really think that being able to use yeast model systems and platforms for discovery, for doing high throughput analyses, and then going back to neurons, and then taking things from neurons and bringing them back to yeast and trying to figure out how they might work, could be used and applied to a variety of other of these protein misfolding diseases. And I'd like very, very especially to thank the students and postdocs in my laboratory who did all of this work, and especially, I've had a wonderful, wonderful, wonderful group of people in my laboratory. I especially thank Tiago Utiro, who tackled the a-synuclein project when not only was everybody from outside labs telling us we were crazy, but the people in my own lab were telling him he was crazy. What the heck are you doing? And Anil Kashikar and Aaron Gittler did all of the high throughput screening on that model. Julie Su worked on the compounds, and these folks worked on other aspects of the project that I haven't told you about. These are the folks that are working on the A-beta model in yeast. And this is our amazing, wonderful, wonderful group of collaborators without whose help we really couldn't be where we are. So I'd like to thank you again for listening.